So diffusion chamber is one of the techniques that is used for, uh, I mean, is a kind of technique to attempt cultivation or culturing of unculturable bacteria, right? So diffusion chamber, it was uh, designed by Lewis and Epstein, and continuously till date there are various advancement in the design of these diffusion chambers, right? So what you are going to do, you are going to entrap these bacteria, or you are going to enclose these bacteria within a semi-permeable chamber. So there is a metal disc, and below and above surfaces are nothing but simple semi-permeable membranes, and entrapped between these membranes are the gelling matrices, uh, along with the source from where you are taking the sample mixed with it. So you are going to inoculate onto that gelling surface, right? And this chamber is then going to be floated onto the mm, what you can say is a small aquarium which you have developed to mimic the actual natural conditions in the given environment, right? So you are going to use this gelling disk so that it is separated from the rest of the environment, because you have put a semi-permeable membrane of a defined pore size that will not allow the movement of bacteria across it, but chemicals, nutrients can pass through and will be made available to the bacteria which are growing onto the surface. So the chambers are inoculated with dilute suspensions of the cells from natural habitat, or maybe something like marine sediment, or the sediment from some freshwater body, whatever your target ecosystem that you want to explore for, right? And then you are going to put this sealed chamber onto the top of aquarium of seawater, or you can say you can prepare a bed of sand, and then you can just cover it with the dome, that is digested organic matter, which is present generally at the base of every water body, that can be layered onto it, and some amount of water just to facilitate the conduction of nutrients across, right? And you are going to float your disc on the top of that, and then you are going to incubate it and observe what kind of growth you are obtaining. So you can just leave that chamber and observe it under microscope, or you can purify the colonies growing onto it, onto spatial media. But again, is it going to appear as actively as it is seen in the diffusion chamber? Are they going to be transferred efficiently and will be incubated and will be growing into colonies? So is the same success expected? Yes, do expect the same colony, if it is growing in the diffusion chamber, should also grow in the petri plate. But that is not the scenario always. Maybe in the in the diffusion chamber you are seeing it, but it may not appear when you attempt to purify it in the petri plate. Why and how to get rid of these things, that we will talk. So comparison of number of growing microcolonies, if you see, generally the recovery rates are very less, and maximum up to 40% people could attain, right? So recovery rate is as less as 0.05%. So hardly one or two colony out of hundreds that you have transferred would survive. Why would this happen? Again, it goes back to the point, that is the microenvironment, or you can say the exact mimicking. I mean, sorry, you are not able to mimic the natural environment exactly, and so environment is not really conducive for the organism to grow. So several of the microcolonies from the chambers can be isolated in pure culture and determined to be previously uncultured bacteria. That is what we expect. But as said earlier, the revival rate could be as less as 0.05%. So, in addition to diffusion chamber, a modified design, you can say, that is referred to as a trap, that also people have tried. In that, membranes enclosing it has a pore size, and it will allow selective type of bacteria to pass through it. Maybe actinomycetes and their filaments will grow, and all non-filamentous bacteria cannot pass through. So these examples we are going to see in the next slides. It was just a overview to reconnect you with the previous lecture. Now, when I talk about this diffusion chamber, this is the design that we had talked about, right? It was redevised in 2002 by Keberlin, and even today many modifications in this design are happening. So we are trying to simulate the natural environment, right? So the chambers are designed, of course they have to be sterile, right? These are going to be inoculated with the source material and incubated under conditions, the conditions which mimic the natural environment. So in the diffusion chamber you have inoculated. From that you will, if you see the colonies, you will transfer it onto Petri plate and you will proceed for isolation. But at the same time, the diffusion chamber is not removed from its incubation conditions or incubation chamber. It continues. So when you trap some of these isolates and transfer to Petri plate, you will get generation 1 isolates. But further incubation will provide you with more isolations, but it will be referred to as generation 2 or generation 3 or generation 4 consecutively, right? You are going to incubate those diffusion chambers for a long time, and at intervals you are going to check for the presence or appearance of colonies, and that you are going to attempt to purify. So every time you take the sample out, it is numbered as isolate number 1, 2, 3, whatever from generation 1. Next time when you make an attempt, it is generation 2, and isolates from generation 2 they are numbered. It is just a method, and to understand how long it takes, or what kind of incubation time it would demand, right? Get a uh, new isolate, or the growth of the colonies which were earlier not growing. Now this is another thing that trap, the design of the trap that we were talking here. So in addition to the diffusion chamber, what you are seeing here, you are going to have semi permeable membranes of different pore sizes, all right? So here you are going to have 0.02 to 0.03 micron membrane. This is the metal disc, right? Another thing that you are going to see is this is another membrane, and in between these membranes you have the gelling matrix, right? And then this is kept onto the surface of the mimicked environment, maybe a aquarium, maybe a rock bed, maybe a soil bed, whatever it is, right? And then you are going to look for the number of colonies that are appearing. Now, only the difference is the semi-permeable membranes, they are of different pore sizes. And the upper cover in the previous case, diffusion chamber, was just a sheet, right? It didn't allow any microorganisms to come out. But here you have defined pore sizes. So based on their size, microbial cell size, specific bacteria or hyphae of actinomycetes can pass through, and non-filamentous bacteria cannot pass through if the pore size is as this as 0.02, right? You are aware of that. So here an attempt is made, and to compare the performance of two different approaches to grow the unculturable bacteria. One is diffusion chamber and another is microbial trap. So these in the when the growth is seen in the diffusion chamber gel, on the top of the gel in the diffusion chamber, it was picked up and was put or transferred onto the Petri plate, and what you get in the agar matrix, a few colonies of the bacteria. But if you try to use a microbial trap and allows that allows the movement of fine structures, microbial structures like hyphae of actinomycetes, they only are allowed to pass through the the pores of the membrane. So these blue uh, blocks, they are nothing but the matrix of the membrane. So here the gaps are nothing but the pores of the membrane. So actinomycetes are growing here, and their filament can pass through 0.02 micron layer. So depending upon the requirement, such modifications have been made in the designs of diffusion chamber and traps, right? This is even higher version, which is referred to as iChip. Introduction related to this uh, we had discussed in the previous lecture also. So different approaches out of this, if you see, here is a metal chip, right? And if you are able to see, there are fine uh, what you can say pores, more than 300 pores are there on this particular small chip, right? And this is the something like you have the strainer at your home that you use for straining say tea, right? So fine mesh is there, but here of course the mesh size is very very small. It is as less as zero point 0.2 0.3 micron depending on the bacterial morphology that you have to choose. And 0.03 micron pore size is the best because you want that bacteria to get retained. If it is as less as zero point 0.3, it will not um, uh, uh, get onto the surface of the media. So the device is going to consist of a central plate on which you are going to have hundreds of pores, right? And you are going to have a membrane which is going to be a 0.03 micron pore size on both the top and bottom of the plate. And these are nothing but the another panel, and these are the screws which you are going to use to tighten this particular assembly, nothing else. All right. So here each pore is going to act like a petri plate, you can say, or a culture vessel, and whatever a single bacteria which is going to get entrapped in that place will develop into a colony sequentially, subsequently, right? So it is possible to isolate as many as 300, 387. In this particular case, the number of pores which are engraved onto are 387. So so many isolates you may get by this kind of a arrangement. So people are making such what you can say the designs to facilitate the growth of unculturables. All right. Now, going beyond these designs, we have to talk about the microbial interactions which are going to be helpful for each other's growth, right? So, when you talk about diffusion chamber, for example, incubated for 1 to 4 weeks, you are going to get the isolated colonies. Maybe diffusion chambers, like they have been used for diverse environments, made be sediments, soil, or marine sponges, the flora associated with marine sponges, and have been found to be highly efficient in terms of microbial recovery. But yet, there are certain challenges that bacteria which are uh, dominant among within a given environment or ecosystem, but when they are tried to be grown in laboratory, in spite of using things like a diffusion chamber or eye chip, etc., are not able to grow because they need some of their specific members of their community on which they are dependent, right? So here uh, certain specialized methods are like eye chip or diffusion chamber, and all these things though are referred to as high throughput methods. High throughput, that is, they are going to give high rate or high success rate when it comes to isolation or purification on bacteria. So they are there, but yet they also will not assure you the growth of certain bacteria which need those have specific requirements, and they can be made available by their companions which are present in the community, right? So what about those? So in that case you have to think about uh, uh, intermicrobial interactions and what kind of bacteria bacterial systems are working, and what kind of give and take mechanism is working among those bacterial system that you need to understand. So I'm trying to share one example of that kind, which is prochlorococcus, which is a lot talked about. Okay, it is known as the most dominant photosynthetic species in the marine environment. So it's going to be accountable for about 50% of total carbon fixation when it comes to photosynthetic bacteria in the marine environment. So, such a dominant species. But when you attempt to grow it in the laboratory, the rate of growth is negligible, rather, it is not really so easily possible. Now it has become possible with efforts over a decade time, and various methods have emerged which could help scientists to understand the requirements of these prochlorococcus strains. Right? As I said, there are a number of prochlorococcus cells per millimeter. It could be as high as 1 lakh cells in 1 millimeter volume of ocean water you will find. This number will tell you how dominant species these are in a given marine environment. And there are different species of it, yet only 5% uh, uh, of species are 5 to 10% species of these are now been cultivated. But the concern is these are the species which are going to be fixing the atmospheric carbon and being chlorophyll containing. Respiratory mechanisms are based on combined with photosynthesis, and you are going to they are going to contribute the carbon content of the seawater. Okay, so see the number of cells per unit volume, 1 lakh per millimeter. So what is the difficulty in making them grow in the laboratory? Let's know this interesting story. So in this case, this MIT 9313, now this is one of the strains that these people have attempted to isolate, is a chlorotic cell. They show reduced metabolic activity measured as carbon and nitrogen uptake by nanoscale secondary ion mass spectroscopy. Why I'm reading this, that you get to know कि when you are talking about ecological studies, you have to take help of various analytical tools. So one of these which we talk here is ion mass spectroscopy for measurement of the metabolic activity in terms of carbon and nitrogen uptake at the nanoscale by individual cells. All right. So when it comes to the survival of this organism, now they are chlorotic species, they have chlorophyll, so carbon dioxide is a essential component for their growth, right? And another thing is axenic conditions, right? So they do not regrow under axenic conditions when they are transferred to new media. So these are the two important points when it comes to discussion about prochlorococcus. Now let's see what this prochlorococcus is, why is not growing in the laboratory. Now are you able to see this particular diagram which shows two prochlorococcus cells, and there are arrows shown here. Right? I'll try to, yeah. Are you able to see these small arrows? They are showing some circular structures within the cells. Alright? Could you notice this? Yeah. One, two, three in these cells. In these cells there are two. And if I and in this particular cell there are no such structures seen. Right? Are you able to notice this? So it is showing you a number of the CO2 harvesting structures within the cell. Alright. So if the atmospheric CO2 concentration increases, this availability of CO2 will be easier for these cells, and these structures which are harvesting CO2, they will reduce in the number. So like this case, so the second figure which is showing the case where very ample or high amount of CO2 is available, and this is the case when CO2 is CO2 levels are less. Okay, now, and a clear cut message is there कि under presence of high amount of CO2, easy availability of CO2 is there, which is essential for its respiration and photosynthesis. But because it is easily available, then the number of structures which are going to harvest CO2 from environment, they will get reduced. Okay. Now here in the second picture you can see two types of bacteria. One is this Prochlorococcus, which is shown in green, and another is shown in orange. This is the second organism is Alteromonas, right? And can you imagine these two different genera cannot survive in absence of each other, and their presence in turn is regulated by CO2, which is present in the air. Now, if you observe the diagram, it will show you that oxidative stress, of course, is experienced by every living cell. So, this bacteria also will experience oxidative stress. But in this case, HIL genes, HIL genes, they are playing a role to reduce the oxidative stress in case of Prochlorococcus. And in case of Alteromonas, mechanism is different. It is producing catalase enzyme, and that catalase is going to take care of all what you can say the oxidative species which are going to get generated and attack on that, right? But the point is cleaning of oxidative stress, you can say a mechanism of cleaning out, so they are different in these two. But what happens when the CO2 levels, they are going to get elevated in the environment? The number of these vesicles which are involved in CO2 entrapment, they are reduced. HIL genes expression also gets decreased in Prochlorococcus. As a result, its mechanism to fight back oxidative stress is compromised. Okay, so it has no mechanism to fight back the oxidative stress. At the same time, in case of these Alteromonas also, its catalase gene expression gets compromised if CO2 levels are high in the environment. So oxidative stress is getting built up in the niche, and this Prochlorococcus does not have a mechanism of its own now to reduce this oxidative stress. Also, this Prochlorococcus upregulates the CCRG1 gene, which is one of its defense mechanism against bacteria that would attack this photosynthetic organism. All right. So overall, this cell is in a trouble condition now, and the Prochlorococcus cells will start dying. Understanding this mechanism, it is very clear to you that Alteromonas and Prochlorococcus, they have interactions, and the interaction is going to support the survival of both these organisms, and their growth or survival are dependent on atmospheric CO2 levels, right? So now, if I have to take help of this information and go for uh, uh, cultivating these bacteria in the laboratory, what should be my approach, right? So Prochlorococcus or Syncoccus, these are abundant unicellular cyanobacteria, and as I said earlier, they are major participants in the global carbon cycle, so their presence is very essential, right? So if they get killed because of increase in the atmospheric CO2, it is going to uh, cause a big threat to the sea ecosystem, all right? But these fluctuations are surely happening when it comes to CO2 levels. But how are they compensating it? How the oxidative stress is taken care of, and how it is affected by CO2 levels? That is what we talked, right? Now, can we take help of this information to make these two different bacteria uh, to grow in the laboratory? So if I am interested in growing Prochlorococcus, I shall find the neighboring organism like Alteromonas, and that should be growing in my plate, right? But the thing is Alteromonas so easily cultivable? No, not at all. then what is that mechanism which of Alteromonas that helps Prochlorococcus to grow is to be understood. Yeah, now I know catalase that is produced by Alteromonas helps Prochlorococcus to combat the oxidative stress. Under high CO2 conditions, it becomes a challenge to have its own stress fight mechanism, like oxidative stress fight mechanism, to be working. So when I am attempting to grow this bacteria in laboratory conditions, see uh, the gaseous environment is altered, right? And it does not have its companion that would provide the oxidative stress reducing mechanism. But I know now the catalase is the one which is required to reduce the oxidative stress. So can I add some of these oxidative stress reducing molecules in the media or make it available to the bacteria in its environment when I'm trying to grow it in laboratory environment under artificial conditions? That can be one of the approaches, right? Or second could be, can I grow the bacteria that produces catalase in the same plate where I am trying to grow Prochlorococcus? So both of them are growing as co-culture, and whatever kind of microbial interaction was happening in the natural environment can be to certain extent mimicked, and give and take mechanism of bacteria of certain chemicals will help them grow, sustain and grow, right?